Another example of a compound classified as a lipid-derived autacoids in addition to eicosanoids is a platelet activating factor. This presentation will talk about the introduction to the platelet activating factor, or the PAF. We are also going to talk about its chemistry and its synthesis, its pharmacologic effects, and the pathophysiological roles of the platelet activating factor. And lastly, we also have the therapeutic uses of the platelet activating factor. The platelet activating factor is another autacoid derived from the membrane phospholipids and is therefore related to the eicosanoid family. The PAF is also known as acetylglyceryl ether phosphorylcholine, or the AGEPC, and it is a cell membrane derived autacoid. Like the eicosanoids, the PAF is not stored in the cells but is synthesized in response to stimulation. So we have here a diagram showing the major sources and stimuli of the platelet activating factor. The major sources of the platelet activating factor include the polymorphonuclear cells such as the eosinophils, neutrophils, macrophages, basophils, mast cells, as well as the endothelial cells and the platelets. The PAF production in the endothelial cells is triggered by thrombin. So the thrombin is an example of a stimuli for the production of the platelet activating factor. For the neutrophils and eosinophils, they are stimulated to synthesize and release platelet activating factor through chemotaxis, or the chemotactic factors, as shown in this figure. The platelet activating factor is secreted in IgE-mediated reactions by basophils and in IgE-mediated reactions by mast cells, as shown in this figure. So the basophils and the mast cells represents the major sources of the PAF, and the stimuli include the antigen and the IgE. So these cells can be stimulated to release histamine and platelet activating factor through a number of biochemical pathways that might play a role in mast cell activation. Chemically, the platelet activating factor is 1,O-alkyl 2-acetyl SN-glycerol 3-phosphocholine. It is derived from this compound that is found in the membrane of many cells. So this diagram shows the structure of the platelet activating factor. For the synthesis of the platelet activating factor, the PAF synthesis involves the action of the phospholipase A2 enzyme on this compound to yield lysoplatelet activating factor. The lysoplatelet activating factor, or the lyso-PAF, is acetylated by acetyl coenzyme A in the presence of the enzyme lyso-PAF acetyltransferase. It is mainly released from many different cells, including neutrophils, activated macrophages, eosinophils, mast cells, and of course from platelets. The action of PAF is not only limited to platelets but also acts on many different cells. So this diagram represents the chemical structure of the platelet activating factor. The platelet activating factor mediates its effects by binding to the PAF receptor, which couples with at least two different types of G proteins, the GIO and the GQ. So the platelet activating factor receptor is a G protein coupled receptor. The downstream effects of the receptor signaling is mediated via the nitric oxide synthesis in the endothelium. For the pharmacologic effects of the platelet activating factor on the cardiovascular system, the platelet activating factor is considered to be a potent vasodilator. It lowers the blood pressure and the total vascular resistance. Secondly, the platelet activating factor increases the vascular permeability and the movement of fluid out of the vascular compartment. The platelet activating factor is considered to be about 1,000 times more potent than histamine or bradykinin. The platelet activating factor is considered to be the most potent agent known to increase vascular permeability. For the pharmacologic effects of the PAF on the blood, the PAF is considered to be a potent promoter of platelet aggregation, and it also stimulates the polymorphonuclear leukocytes and monocytes to aggregate. For the smooth muscles, although the platelet activating factor lowers the blood pressure due to its vasodilatory or relaxing effect on the vascular smooth muscle, it markedly contracts the smooth muscle of the gut, stomach, uterus, and the peripheral airways of the lungs. Let's now proceed to the pathophysiological roles of the platelet activating factor. The PAF is a mediator in a number of physiological and pathological events in the body. Derangement and dysregulation of PAF functioning has been associated with some diseases. PAF has pro-inflammatory actions, or it promotes inflammatory reactions. The PAF is elaborated by leukocytes and mast cells at the site of inflammation and appears to participate in the causation of vasodilation, vascular permeability, and exudation, cellular infiltration, and hyperalgesia. So when we say hyperalgesia, that refers to an increased sensitivity to feeling pain and an extreme response to pain. The PAF also has a role in allergic responses and anaphylaxis. So when we say anaphylaxis, that refers to a severe, potentially life-threatening allergic reaction. The PAF is reported to be involved in allergic and anaphylactic reactions. It appears to play a major role in bronchial asthma, along with leukotrienes. It causes bronchoconstriction, mucosal edema, and secretions. High circulating PAF levels are associated with anaphylactic and shock conditions. PAF stimulates platelet aggregation and promotes hemostasis and thrombosis. So when we say hemostasis, that refers to a process to prevent and stop bleeding. When we say thrombosis, this occurs when blood clots block veins or arteries. The clot is known as a thrombus. So in this aspect, the PAF does not function independently but contributes in a manner analogous to the action of thromboxanes and adenosine diphosphate. PAF also has a role in reproduction and parturition. It may be involved in ovulation, fertilization, implantation, and parturition. In the absence of PAF, it is said that ovulation does not occur. Early embryos which produce PAF have a greater chance of implanting, and PAF produced by fetus at term may help in the initiation or progression of parturition. PAF may be involved in progression of labor by directly contracting the uterus and releasing PGE2. At the time of parturition, PAF aids in increasing uterine contractions. And just before parturition, The compound is found in the amniotic fluid, that is, it is released from the fetal lungs. For the drug stimulating the activities of the platelet activating factor, so unlike some eicosanoids, the stimulation of the platelet activating factor activity serves no therapeutic response. The platelet activating factor or its analogs has no pharmacological or therapeutic roles. For the inhibitors of the platelet activating factor receptors, the PF receptor antagonist, lexipafant, has recently been used for the management of acute pancreatitis. The anti-inflammatory property of glucocorticoids is in part attributed to the inhibition of the platelet activating factor synthesis. Another group of autacoids that is classified as a peptide autacoid are the cytokines. So what are cytokines? In order to mount and coordinate an effective immune response, a mechanism by which lymphocytes, inflammatory cells, and hematopoietic cells can communicate with each other is required. Cytokines perform this function. Cytokines are a family of small proteins that mediate an organism's response to injury or infection. They operate by transmitting signals between cells in an organism. Minute quantities of cytokines are secreted, each by a single cell type, and regulatory functions in other cells by binding with specific receptors. Their interactions with the receptors produce secondary signals that inhibit or enhance the actions of certain genes within the cell. Unlike endocrine hormones, which can act throughout the body, most cytokines act locally near the cells that produce them. The two principal producers of cytokines are the helper T cells or the Th cells and the macrophages. They both influence the innate and the adaptive immune responses. On the right shows the mechanism whereby cytokines are produced from cells and their mode of action. So in the presence of a stimuli, the cytokine producing cell will produce a specific type of cytokine. These cytokines will act locally and it will bind to its receptor on the target cell. The binding of the cytokines to its receptor will produce a signal and will result in the activation of the genes, and the gene activation will produce a biological effect of that particular cytokines. The diagram on the left shows the specific examples of cytokines and their action. So one of the example of cytokines are interferons. So interferons are antiviral proteins. We also have the chemokine family. So this group of cytokines have a function on direct cell migration, adhesion, and activation. We also have the TNF or the tumor necrosis factor, which are important in the regulation of inflammatory and immune responses. We also have the interleukin. The interleukins are a variety of actions dependent upon the interleukin and cell type. For the hematopoietins, these type of cytokines promote cell proliferation and differentiation. For the transforming growth factor beta family, they are important in the regulation of immune cells. Cytokines are considered to be pleiotropic. So when we say pleiotropic, this is a property wherein the compound will act on different cell types. They are also regarded as redundant, meaning that the different cytokines carry out the same function. And they are also said to be multifunctional, meaning that the same cytokine can regulate several different functions. So a while ago, we have enumerated specific examples of cytokines. So for example, examples of cytokines are tumor necrosis factor, TNF, interleukins, as well as the interferon. So it is said that there are three functional categories of cytokines. So the first category are those cytokines that regulate the innate immune responses. So basically when we say innate immune response, this is also referred to as the innate or the non-specific immunity. So this is the defense system with which the individual is born. So it protects the individual against all antigens. The mediators and regulators of innate immunity are produced mainly by mononuclear phagocytes in response to infectious agents. We also have those cytokines that regulate the adaptive immune responses. So when we say adaptive immune responses, they are also known as the adaptive immunity or the acquired immunity. And this involves specialized immune cells and antibodies that attack and destroy foreign invaders and are able to prevent disease in the future by remembering what those substances look like and mounting a new immune response. The mediators and regulatory of the adaptive immunity are produced mainly by T lymphocytes in response to specific recognition of foreign antigens. We also have those cytokines that stimulate hematopoiesis. For the nomenclature of cytokines, so it is said that there are two major groups of cells that are capable of producing cytokines, and they are the T helper cells as well as the macrophages. But there are a variety of cells that can produce cytokines upon proper stimulation. So the cytokines released by lymphocytes are called as lymphokines. We also have the monocytes and macrophages. So the cytokines that are released by monocytes and macrophages are known as monokines. We also have those cytokines that are released by leukocytes that act on other leukocytes are referred to as the interleukins. The cytokines that are released by cytokines with chemotactic activity are called chemokines. So it is said that there are two principal producers of cytokines, the helper T cells or the TH cells and the macrophages. Although they can be transiently induced and secreted by virtually all nucleated cells. So the cytokines that are released by a mature helper T cells, so these mature helper T cells are classified as lymphocytes. So the cytokines that they release are called lymphokines. The cytokines that are released by macrophages are known as monokines. This diagram shows some important specific examples of cytokines. As shown in the figure, some important cytokines include the interleukins 1, 8, 6, and 12, as well as the tumor necrosis factor alpha. They are derived from macrophages. The macrophages are stimulated to secrete cytokines by the bacterial lipopolysaccharide derived from the outer surface membrane of the gram-negative bacteria. In the presence of gram-negative bacteria, the macrophage will ingest and degrade that bacteria, and it will be activated by the bacterial lipopolysaccharide to secrete cytokines. Some of the cytokines that will be produced are the interleukin 1. So the interleukin 1 will produce local effects as well as systemic effects. The same is true for the other interleukins as well as the tumor necrosis factor alpha. To cite an example of the local effects that are produced by the interleukins, particularly the interleukin 1. So the interleukin 1, upon binding to a specific receptor, will result to the activation of the vascular endothelium, activation of the lymphocyte, local tissue destruction, and increased access of the effector cells. The interleukin 1 also has its systemic effects, and that is the reduction of fever together with interleukin 6. We also have another type of interleukins, particularly the interleukin 6. So this interleukin 6 will result in the lymphocyte activation and increased antibody production. For its systemic effects, so it will induce fever, induces acute phase protein production. For the tumor necrosis factor alpha, so it also has local effects and systemic effects. So when this particular cytokine will bind to a specific receptor on the target cell, it will induce the activation of the vascular endothelium and increase its permeability, which leads to increased entry of the immunoglobulins, the IgG complement and cells, and increased fluid drainage to lymph nodes. For its systemic effects, it will induce the production of fever, as well as the mobilization of metabolites and the induction of shock. So those are some of the local effects and the systemic effects that are produced by these specific types of cytokines that are produced or that are derived from the macrophage.